Hi, everyone, and welcome to the September meeting of the Linnaean Society of New York. I'm Debbie Mullins, the president, and I'd like to call this meeting to order. We're so glad that you're able to join us for the first membership meeting of the new year. Our Vice President, Doug Futuma, has put together a great lineup of speakers for this year. I've had a sneak preview of tonight's presentation, and it's going to be really interesting. If you haven't sent in your dues yet, please do so as soon as possible. You can renew your membership online using a credit card, or you can pay by check. Please take a minute before you turn off your computer tonight to pay your dues. Just go to linnaeannewyork.org/members. Our annual homecoming picnic is a week from Sunday, September 24th. Please sign up soon if you have not already done so. You can register on our website at linnaeannewyork.org/homecoming. This year we'll be at the Heckscher Playground picnic area at the south end of the park. Please bring your own sandwich and we'll provide snacks, sweets, and beverages. Last week, you received an email from me with information about the Lights Out bill, Intro 1039, that will be voted on by the City Council, and the Birds and Bees Protection Act that is awaiting Governor Hochul's signature. These two pieces of legislation have the potential to prevent avian mortality. So if you haven't written those letters yet to your City Council representative and to Governor Hochul, please do so soon. We have the results of the Great Gull Island Birdathon. Last May, many of you participated in the Birdathon or sponsored one of our teams. We had a really great result this year, raising $5,800, more than twice the amount of the previous year. Again this year, the noble gnatcatchers won the species count with 116 species, but the unchained bobolinks were close behind with 106 species. The Saturday morning slow birders group raised the most money, bringing in $2,295. Many thanks to Michelle Zorzi and to Debbie Becker for organizing the Birdathon and to everyone who participated or sponsored a team. And speaking of Great Gull Island, our speaker at the October meeting is Dr. Margaret Rubega, who will be talking about conservation planning for the terns on Great Gull Island. So you'll be sure not to miss that. Last week, you were asked to vote on two motions, one to approve the minutes of the May membership meeting, and the second to approve the membership applications of new members. The minutes of the May meeting were approved by a vote of 126 in favor and 2 abstaining. And I'm pleased to announce that 23 new members were approved by the membership as 23 people were approved by the membership as new members. The vote was 128 in favor and none opposed. Our membership now stands at over 700 people. Our new members are Ruth Ryu, sponsored by Meredith Barchett and Russ Como, David Schneiderman, sponsored by Ann Lazarus, Robert Kennett, sponsored by Ken Chea, Richard Devereaux, sponsored by Barbara Saunders and Nancy O'Keefe, Noel Comas, sponsored by Michelle Zorzi, Nancy Tollefson, sponsored by Kevin Sisko, Barbara Mahoney-Kent, sponsored by Kathleen Howley, Lisa Curtis, sponsored by Karen Becker, Ann Woodhouse-Plum, sponsored by Mary Beth Cooper, Timothy Ryan, sponsored by Philip Ribelow, Yuri Shim, sponsored by Karen Asakawa, Tamar McKaylee, sponsored by Amanda Bielskis, Diane Louie, sponsored by Gabriel Willow, Darlene McNeil, sponsored by Doug Futuma, Gary Himes, sponsored by Ken Chea, and Ann Himes, also sponsored by Ken Chea, Rachel Siegel, sponsored by Mary Beth Cooper, Jeffrey Liu, sponsored by Mary Beth Cooper, Michelle Yan, sponsored by Barbara Saunders, Susie Dipple, sponsored by Karen Becker, Sonia Schmid, sponsored by Mary Beth Cooper, Marlene Panton, sponsored by Ken Chea, and Amy Davidow, sponsored by Mary Beth Cooper. I extend a warm welcome to all these new members. We're very happy to welcome you into the society, and I hope you'll join us at the homecoming picnic so that we can meet you in person. If anyone listening tonight is not a member, please join. The membership application is on our website, linneannewyork.org/members. And don't worry if you don't know anyone to ask to be your sponsor. I would be happy to sponsor you, Debbie Mullins, as would any member of the board of directors. We value the diversity of our membership and encourage everyone to join. All you need is a genuine interest in natural history and a desire to explore those interests with like-minded people. So now let's turn to tonight's presentation. We've disabled your microphone and the chat function, so please use the Q&A feature to submit questions for Dr. Dávalos. Following her presentation, Vice President Doug Futuma will moderate a question and answer session. So tonight we're very happy to welcome Dr. Liliana Dávalos to the Linnean Society of New York. Liliana is a professor of conservation biology at Stony Brook University. Her focus is on biodiversity and conserving the world's life support systems into the future. In her research, she uses genetics, genomics, and statistical tools to discover mechanisms of extinction and survival. Dr. Dávalos received her Bachelor of Science degree in biology from the Universidad del Valle, Cali, Colombia, and a PhD in ecology, evolution, and environmental biology at Columbia University. She's had a distinguished career, having published over 90 research papers in leading scientific journals. She is a 2012 National Academies of Sciences Education Fellow in the Life Sciences and a 2013 Kavli Frontiers of Science Fellow for an outstanding early career. She has advised the United Nations Office on Drug and Crime on Deforestation since 2017, and she's a member of the Science Panel for the Amazon. The title of her presentation tonight is Phyllostigmid Bats or a Unique Mammalian Radiation. Here's why. So Dr. Dávalos, welcome to the Linnean Society.
Thank you so much for this wonderful invitation, and thank you so much for just a wonderful introduction, inviting me to give a talk, and of course for coming to it. There's a slight tweak in that when I put the abstract together, it was about phyllostigmid bats, and I'm going to kind of expand that group a little bit to talk about Noctilionoid bats. I was also very encouraged because at first I was seeing that all the pictures were of birds, but I noticed that you have one red bat in among the pictures, so I can see that my audience is going to be interested in the bats as well. But since they fly, I hope that we are all looking forward to this, to looking at the flying mammals of the night. Okay, so to give you an idea, so when I was invited to this, I kind of wondered how I would present things to an audience of the Linnean Society. And so today's talk is structured in 3 sections. They're not all equally long, but roughly, first I'm going to talk about looking at the past. And when I say the past, I'm talking about fossils and ancient extinctions and extinctions that are happening closer to the present. Then I'm going to talk about how these Noctilionoid bats, which include the phyllostigmid bats, how did they get so diverse? How did it come to be that there's so many different species of them and they do so many different things? Then I'm going to talk a little bit about the genes that are involved in some of the things that these bats can do, this particular bats can do that we can't, we as humans. However, I always feel like a talk like this, if it's just fact, and stories about the animals, it also needs a human dimension, a dimension of how the science gets done. And so there's going to be these 3 themes as well that are intermeshed with the scientific finding. And in the first section of the talk, I'm going to talk about serendipity, the opportunities that just kind of come when you're prepared or you don't really know where the next thing is going to come from. And then serendipitously, you find this big interaction that it's really good. Then I'm going to talk about persistence. So a lot of us in science, we have to persist at the questions because what we decide to take on is often very difficult to do. And there's a reason why people haven't done it before, and it's because it's hard. And so it's important to persist at asking the questions that you're interested in. And finally, I'm going to talk about resilience, because one of the things that happens in science is that sometimes things don't go the way that you think they're going to go. And so you got to be able to bounce back from that. And I'm going to talk about that as well. So without further ado, I'm going to start with looking at the past to protect the future. And when I'm talking about the past and the future, I'm talking about bat diversity. So I'm going to give us a little bit of a moment to have a look at these lovely faces. And I hope that you agree with me that these are really amazing diversity. Most of us, we're familiar with birds because we can see them in the daytime and we can see their colors. But with bats, it's different. They're flying at night. And so only the proportion of people that can actually appreciate what they look like is a lot smaller unless we have access to a picture like this or to a book, where we can see that immediately. It's striking how sometimes you have these very large eyes, right? So even though they're flying at night, sometimes bats can have very large eyes. Sometimes they have tiny eyes. Sometimes I like to point out there are some bats. So if you look at bat number 20, the eye is actually kind of on the inside of the ear. So you see the structure of the ear is coming outside and the eye is kind of right in the middle of it, right? And so typically we tend to think of bats as number 8 with this very, very big ears, right? Having big ears. But now in this picture, number 4 has gigantic ears. But if you look at number 30, right, like you see this Pteropus, it's got like really, really tiny ears. So we have this huge amount of variation that's already cluing us about what that diversity is like and what they might be doing different. In amongst this huge diversity, so these are bats from all over the world that encompass all of the groups of bats that live to this day. In among this huge diversity, there's one group that's particularly diverse, and that group is called the Noctilionoids. So when we study bats, most of us don't focus on bats in general, but some bats in particular. This is called a superfamily, and it's got some highly popular in the world of bats. So number 2 is Noctilio, the fishing bat. So it's one of 2 different species of bats that are capable of fishing fish out of the water and eating from them. Number 3 is a vampire bat, right? So there are only 3 species of vampire bat, and they're the only vertebrates that are capable of feeding exclusively on blood. And they are in this part of this superfamily. We got number 20 that we already talked about with the tiny eyes. Number 26 that looks like a beautiful cotton ball that's found in the forest, the Honduran white bat in the forest of Costa Rica. It's a really, really tiny, tiny bat with yellow ears. Number 25, Lone Corina. That's a really special bat. You can see that the nose sleeve is extending out of its face, and we still don't fully understand what it's doing. Number 19 is the largest bat of all the Americas. Vampirum spectrum, it's called. It's the largest of all, and we'll see a picture of later of what the skull looks like. But there's also diversity in the diet. So number 21 is a nectar feeding bat, as you can see. So if we were actually going to take the phylostomids, which is the group that I originally wrote the abstract about, all that it does is that there's 2 fewer bats in this picture. So you can see that the bulk of the diversity of the Noctilionoids, which I'm going to switch between Noctilionoids and phylostomids back and forth, the bulk of all this huge diversity is actually in the phylostomid bats. So talking about this Linnaean society and thinking about natural history, it takes me back. It really takes me back all the way to the beginning, before I even was in graduate school. So to take a look at what that was like. So this is me like in the summer before I came to graduate school in New York, in a river on the western Cordillera of the Andes in Colombia, right? And I was about to move from where I'm originally from in South America all the way to New York City to work at the Museum of Natural History. I didn't know exactly what topic, but there's also a little bit of serendipity or sort of opportunity in that originally I actually wanted to work on birds and I sent an application in. But I think the person who was in charge of looking at the applications from the bird side was busy that year, or they were away at the time when I submitted my application. So this is how it landed. My CV actually landed with the bats, which it's not what I was applying for, but it's actually what I got. And so my office at the Museum of Natural History was actually close to this entrance on 77th Street when I began. And so one of the questions that first comes up when you enter graduate school to do this kind of work is, well, okay, so what kind of questions you want to take on and what part of the world you want to work on? And so I picked, based on what seemed to be the biggest questions that were interesting to me at the time, I decided that was what I was interested in was actually this region of the world, which is the Caribbean islands. So what I'm showing in this slide is that in dark is the outlines of the islands as they appear today. And in this lighter color is the way that the islands were about 12,000 years ago, when the ice sheets came all the way down to Long Island and they took up so much water that they exposed a big area of these islands. And I was interested in these processes because I was interested in answering the question, how did these islands come to have the bats that they have? Because up until that point, there were a lot of ideas about how they had done that, but there wasn't a lot of systematic work that could illuminate exactly how. So over the course of the talk, we're going to see a couple of phylogenies, and some of you may be familiar with them, but I'm going to sort of do a cartoon of how to interpret these things. So for example, if we're hypothesizing that the bats on the island of Hispaniola, which is where the Dominican Republic and Haiti, the two countries that share the island, is, if we think that the bats from that island actually came from Central America, then we hypothesize that there's been some kind of dispersal event that's moved there. And the pattern that we expect, if we think that this is the way things happen, is a phylogeny like this, where there's a species on the continent that's a species A, and it's got a close relative on the island. There's more to it than that. I'm not giving a full picture yet, but the point of it is that in order to answer the question that I was positing of how did bats get onto these islands and why are some bats on some islands and not others, the way to answer it was to generate this kind of evolutionary tree of relationships between all the different species that were found. But there was a problem, right? So if it's a matter of just taking all of the phylogenies or evolutionary trees like the cartoon that I'm showing here in orange, then that would be easy. We just take that and we kind of do some sort of a statistical analysis and come up with some answers. The trouble is that at the time when I started that work, there was no very good phylogeny, or rather the phylogenies that we had were too poorly resolved, right? So what do I mean by that? So let's take a look at this. So as you can see, there's many more things going on than in the previous one. In the previous one, we only had two species, A and B. Now we have lots and lots of species, and I've highlighted all the species that are present in the Caribbean. So these are all the phylostomid bats that are present on the islands today. These are all living bats. But we can see that when we see this two lines back here, Glossophaginae and Brachyphyllinae, we can't really tell who is more closely related to whom. They're not resolved. They're not fully resolved. So since we're traveling down the memory lane, I can take you back on a journey of what was necessary to actually try to answer this question. And it's a mix of really fun things, at least for a graduate student, going in the field and doing things, and a list of a lot of work that I'm not going to get into that. So this seems like the ancient times now that we have so many different ways to sequence, but I actually wanted to show this picture of what that was like. So what that was like, if you see that ladder of colors over here, it's a fluorescent marker. So what we would do is we would take little pieces of DNA. Well, we would actually get a piece of sample from the animal and then extract the DNA, then get little pieces of DNA and multiply them using polymerase chain reaction, or PCR, which you may have heard of for like diagnostic tests. And once we have those little pieces, we actually kind of try to put a fluorescent dye we try to extend it to the length -1, and then the length -1, and then the length -1, and over and over and over until the extension of all the end was marked by one of these fluorescent dyes. And those fluorescent dyes would correspond to A, T, C's, and G's that encode the DNA. And the reason why I'm saying this is ancient history, and I don't know how many people who are doing this work are seeing the talk, but it's really so amazing because back then, there was like this toxic gel that we had to use and we had to tap on it and everything was very difficult. And the same amount of work that I did over the course of something like 4 years, like I could do it here in my basement this evening after the talk and it would be better, like it would be better quality. And that's how far things have come. Is that now we have way more data. But this is the way it was done. And it was a lot of fun because we got to see things like this bat in Jamaica. You can see that there's pollen all over its face. It's been on a bat. And I got to see really beautiful places like the karst beaches that are ubiquitous throughout that part of the world and find amazing bats. Okay. So this lasted a bunch of years trying to, catching bats in different places, in different islands and having a good time that way. And then coming back and doing this sequencing using these ancient technologies by now that are completely superseded. So what did I find out? Well, okay, so in the end, what ended up happening is when we look at the Noctilionoids, which I'm showing here, in the end, what my work ended up being about was the fact that a lot of the events that are leading up, so these events are leading up, they're capitalized here like E, F, G, etc. It's not really too important to get bogged down on them. But these events seem to coincide to periods during which the sea level was low. That's what it ended up being about. Was not necessarily something that I expected originally. When I started doing this work, I thought there would be something more to it than that, but that's what ended up coming out from the data. And in hindsight, getting back to the picture of what's happening in the Caribbean, it kind of makes sense because if we, instead of just looking at the 2 cuts of sea level like we looked at the previous time, if we look at the bathymetry, which is how deep the sea is in the islands of the Caribbean, now we can instantly see that there are these regions that are a lot shallower, so that if there's not just even a glaciation from the last 100,000 years or so, but actually ancient events in which the ice sheets became larger, a lot of land is going to get exposed. And if we have periods of warming like the one we're in now, although right now we're in an extra warming, but anyway, so we can talk about that later. Then the islands become separated. So the point of all of that work ended up being that there is this geological aspect to it that plays a role on where the bats are. I wasn't very particularly satisfied with that. I wrote a bunch of papers. I did a bunch, I try to give talks about it, but I wasn't particularly satisfied because it seemed to me like there was something missing. If we're talking only about how geology imposes itself on biology, then we're kind of discounting what's actually happening biologically. There's also a much bigger picture than that. So if you think about it, this is pretty ad hoc, right? Like the sea level, when you look at the levels of the sea through time and you talk about it with a geologist, they're going to tell you that the sea level is constantly changing. Over the periods of time of the eons, you actually have giant fluctuations happening over and over, such so that if you want to find a pattern there, you're going to find a pattern. So I was more interested in trying to get back at some bigger picture of this, that there is a larger context, and that larger context is actually 60 years old right now. This larger context is a theory called the theory of island biogeography. And to summarize something that, a lot of you may have heard of in the past, what the theory of island biogeography says is that the number of species on an island is going to be determined by 2 rates that you can see here depicted in the 2 colored lines. And so those 2 rates are the rates at which new species immigrate onto the island. But there's this offset that is the red line. That's the rate at which the species go extinct on the island. And whereas the rate of migration onto the island is given by how close it is to the source of the island, like Central America or South America in the case of the Caribbean, the rate of extinction on an island is given or is heavily influenced by the size of the island. so that we can have a prediction about how many species there's going to be on the island, which I was very interested in because some of the islands have lots of species and they're very diverse ecologically, and some of the islands don't have a lot of species. So this larger context was super interesting to me. Unfortunately, even though I had these phylogenies and I had come up with these phylogenies, the methods that are necessary to try to apply this framework to the kind of evolutionary tree that I had not really been advanced. So this is, I think, one of two birds that I'm going to show in the whole thing, and it's a banana quid. You guys are probably very familiar with. And this is one of the methods that had been invented all the way back in that time. So what's this method? All right, so what's happening is Bob Rickliffs and Elridge Birmingham, and they're both great biologists. They had implemented this method, but they had only taken two different ways of looking at it. So over here, we've got the immigration of new species, the new species are arriving, and the species extinction. We don't have anything in this model about, for example, generating new species, evolving. And this evolution can happen in two different ways. It can be that they differentiate in place, even though they stay one species, or they can separate and become more than one species in one. So this model is very simple. It just has the colonization. It just has this two rate, right? The rate of extinction is in the red, and it's pushing the number of species down. And the rate of migration or immigration or colonization is going up, and it's pushing the numbers up. So they use this data from population of birds in the Caribbean to try to fit those lines of when the birds have arrived, fit those little points, put those data points. And they reached the conclusion that they're not finding this equilibrium, this prediction of equilibrium, which is given by the line. It's not what they find. You can see that the points deviate significantly from the line. So because there's no equilibrium, you cannot really have the kind of generality. So for years, after I had done this dissertation, which itself was the product of serendipity in a way, I did not have a toolbox that enabled me to do this. For example, if I did this kind of analysis with my data, it didn't work so well because my bats, unlike these birds, my bats, a lot of them became a different species. They evolved and became either a different species in place, or they actually split into more than one species. So this model did not contain everything that I needed to describe what was happening. And so this is where serendipity really comes in. In 2015, and this is a picture that I'm sure Doug will recognize from the festival that happens there every year, there was a meeting in Bayreuth in Germany. And at this meeting, I was presenting something that looks like that target, which is a much, much bigger phylogeny. So I had continued my work over the years, very focused on the phylostomids and noctilionoids, and I had extended it. But I had this disquiet about what we can do when we had so much data and so much information about the bats of the Caribbean. And Luis Valente, who is shown here on the left, he was presenting there, and he says, well, Liliana, if you actually have all of this information for the bats of this island, I have created a model that overcomes the limitations of the model from 2001. And he and Rampalatian, who is shown to the right, we got to work together with the data that I had from the bats so that we could, for the first time, answer, is this following a process of equilibrium where there is an equilibrium in between the extinction process that is happening naturally over time and the immigration of new species and the speciation process that is happening on the islands. So we were able to do this. So now, as before, we take those different ages where the different bats branch out, and those correspond to those big dots. The big dots correspond to arrival of different bat species. And it includes all of the data that we had for the Noctilionoid bats of the Caribbean archipelago, all of the information about error rates that are associated with them, and also all of the information about speciation. And lo and behold, this was actually, well, we published this a while back, but what was really fascinating about this is that for the first time, by taking all of these data together, we were able to propose that there was an actual equilibrium. So instead of having a situation as with the birds where there was this equilibrium, we actually found that there was a very strong tendency towards equilibrium in the millions of years during which the bats colonized. However, because 13 of my species had gone extinct, so you can see one of the fossils is shown here just as a skull, and it's missing a piece because there was a piece that was missing from the skull that describes that species. Because of that, because of those 13 species, we would actually use our equilibrium model to predict how long would it take for those 13 species to be restored through the natural process of colonization and speciation. And the answer was that it would take something like 8 million years if we had to sit around and wait to restore the diversity, a lot of which those 13 species seem to have disappeared with the arrival of humans on the islands, which happened on different islands at different times. And this was an astounding result because it's the first time that a model of this sort had been used this way. It was the first time that it showed an equilibrium over this eons of evolutionary times, and it was the first time that we could use it to predict what happens after there's this human wave of extinction. Because the Caribbean, you may not know about it, but it is the hottest hotspot of extinction of mammals in the entire world. And the reason why most of us don't hear about it is because these species are already extinct. The islands of the Caribbean used to have its own species of monkeys, like this one that's shown here in every reconstruction. Multiple species and several genera of ground sloths, like this ground sloth that's shown here. Multiple species of rodents called hutias, of which there are only a few species left, and most of which are threatened. A giant vampire bat that's shown inside the trunk of the tree, that's a bat, and it's a giant vampire bat from Cuba that also went extinct around the time of human arrival. And then there are these insectivorants like the Solenodon that actually survived, but the majority other species that were of insectivorant did not survive. So the terrestrial fauna of the Caribbean has suffered some of the greatest extinction waves that we know of anywhere in the world, and a lot of it is associated with people. So what can you do? On the one hand, I'm showing that over evolutionary time, there has been this pattern that these 13 species that are missing would take millions of years, and that there was a tendency in these communities of bats towards an equilibrium where there was immigration from the continent and natural process of extinction. But onto that natural process of extinction, we have this signal of the human wave of extinction. So what that translates for people like me working on conservation biology is that we need to do something about the species that are left in the Caribbean, the species that are endangered. And we identified, together with teams from an organization called Bat Conservation International, we helped identify what were the hottest hotspots where we still had bat species, but the bat species are actually being threatened. And one of those hotspots here is in Jamaica, and this is where this picture is, working together with people from NEPA, which is the environmental organization of the government of Jamaica, Bat Conservation International, plus a couple of scientists and explorers, people who actually explore caves. And we had a pretty good indication because of the past work that the government organization in Jamaica had done, that we would be able to find a species that had not been seen by scientists from outside of Jamaica, had not been seen for more than 40 years. So it was pretty abundant in collection. It's called the Jamaican flower bat, but only the people from NEPA had seen it before. So what does it mean to try to kind of look for a bat like that? You can see that it's a pretty big team and we needed guides because we get into a place called the Inferno Passage. And the Inferno Passage is in this cave that is St. Clair Cave, that is probably one of the greatest places for diversity of bat species in the Caribbean. You can see that the access to the place is rappelling in this case through the ficus, but we can also put rappels on that. However, the problem was that this area of the cave was actually already overrun with feral cats, right? And these feral cats impose their predation on the roosting bat. So even though it's the most diverse cave, right, we already see that the threats are right there in place. And what does it mean to look for bats? Well, this is the Inferno package. So before masks were cool, I was already on a respirator mask in the middle of an underground water that's covered in guano, and the guano is reaching up to our thighs, right? But we're in these waders. And so we are wading through this, looking for the bat species, 2 bat species actually, that had not been seen by other scientists for decades, and looking for that. At another cave where we were working, again, confirming the identification of the Jamaican flower bat, things were a little bit easier and didn't require quite the same level of, let's say, respirators and all of that, because we could be outside of the cave and looking at the bats. And after all of this effort, Bat Conservation International is successfully implementing a program for the protection of the caves, so that the Jamaican flower bat, which is a critically endangered species that is known from fewer than 200 individuals and from only one locality, can persist and can survive in its natural habitat and as part of an ecosystem that includes plants that it pollinates. So all in all, even though I started, this is a story of serendipity in terms of science, of being able to team up with people that could do the things that hadn't been invented yet, but also being able to take action so that this species doesn't become another casualty in the wave of extinction that has engulfed the Caribbean. So now I'm going to talk about how Noctilionoids got so diverse, or how we think that Noctilionoids got so diverse over time. So to remind you, the Noctilionoids include really fun bats like fishing bats, Honduran white bats, vampire bats. And when I say that they're diverse, they are very diverse. So you can see these are all to scale. The skulls are from the Museum of Natural History. The one at the very top right, that is Vampiron spectrum. And you can still see the birdshot, because this bat is so big that the way that specimens were collected was actually with birdshot on the wing that were collected. But at the same time, on the left, on the extreme left, on the top, we actually see this very elongated skull that is from Platalina, which is a bat that's a specialized nectarivore. The 2 rows at the top are phyllostomid leaf-nosed bats, while the row at the bottom is the Noctilionoid bats that I keep mentioning. That includes Noctilionoids that are not phyllostomid. So in general, and as I hinted at the beginning, we tend to think about 2 extreme explanations. Of course, and actually Doug is the author of many a paper about this. There are some things in between, but the idea is that there's a big hypothesis about geography being the one driving the speciation. So a little bit like the story of the sea levels, where we expect this spatial pattern that we have separate places, and the time pattern is that the barriers correspond to the separation between species. So that we expect that if we have 2 species, one a circle and one a square, then the timing in which they separate is the timing in which some kind of barrier, like a mountain ridge or a river, emerge. At the other end of the spectrum, and there's a lot in between, but I'm just going to go to the extremes to make it easier. The other model is that there's ecology, that there's ecological specialization. The spatial pattern, it can be that they can evolve in the same place, or they can evolve in different places. It gets complicated real fast. But what's important to follow is that instead of having this pattern of separation between 2 species by a geographic barrier, we have a pattern in which the 2 species don't resemble one another anymore, because ecologically they're each doing their own thing. Like, for example, imagine a bat that ancestrally it goes around eating insects, but then somehow one group, one population starts eating fruit, and then another population starts drinking nectar. Then they end up like this circle that is white and another black, and they have this fast nonlinear divergence. It's a time pattern that is expected. So we have these 2 extreme models. There are things in between, but we're just going to consider that oftentimes there are people on one side, or there are biologists on one side thinking, yeah, geography is super important, and there are people on the other side thinking ecology is very important. And the Noctilionoid bats are actually fantastic, a fantastic model for answering this kind of question, because they do have a diversity of diet. So, for example, we have blood frogs, we have trichops, which is a frog specialist bat, and blood feeders. We've got the fishing bat, which is pictured here in orange. We've got bats that are primarily nectar feeders, and we've got specialized fruit feeders. So even though they are all part of a single family and superfamily, they seem to have covered so many different dietary niches that this has been a perennial question on the biology of bats, trying to answer that. So this is that target that it looks like it's ready for target practice that I was showing around. So showing all the species that we could get our hands on from all the Noctilionoids, and having all of these species in this evolutionary tree is super helpful, because we can then try to work backwards from these divergences and to see which model might be right, the one that's more geographical or the one that's more ecological. So to give you an example, this is a bird. I didn't show the bird. This is a white eyes. So a bunch of years ago, an acquaintance of mine, Rob Moyle, he did this analysis with this white eyes. And what he found is that if you look at the geological time, Miocene, Pliocene, and Quaternary, is that this clade B, this group, it's just got a huge accumulation of species happening very quickly. So the way he interpreted it was that something special was happening. The diversity builds up. The Quaternary is, of course, the period that corresponds to the glaciations as well. He and his collaborator didn't highlight that role, but if we actually have a geographic pattern, we do expect that there's just going to be this diversity that's going to bump up with the geographic pattern. So what does this pattern look like for my bats? That, as I mentioned, we have this two extreme. What does it look like? So this is the nonlinear accumulation, and you can see on the left is what it looks like for the bats. And you can see that instead of being this huge nonlinear accumulation, like the one that they found, instead we have sort of a pretty linear accumulation over time with some nonlinearity at a different time. So we could, after doing a bunch of statistical analysis, we could actually pretty much rule out the idea that some very recent events have led to the diversity that we had seen in these guys. And instead what we saw was that the rates at which the species were accumulating, they actually are separated into two groups. One group is shown in the warm colors, and the rest, they're not really a group. There are many different groups, and they are shown in the cool colors in the blue. And so the group that's in the warm colors is shown in the dark, and you can see that the inferred speciation rate, and we can have conversations about that, it's significantly higher. So it's always higher than the rate that's inferred for the others. This is a rapidly diversifying group of phylostomic bats that we are seeing. What is different about them, you ask? And the answer is ecology. That's what's different about them. And this is great because, again, we're getting back to the point of not having geography and geology dictate what happens to the biology, but it's actually the ecology. So here's pictured and Artibius, which is an iconic bat of Central and South America, right? And what these bats have done that the other bats don't do is they are increasingly ever more reliant on plants. We can see that on these plots on the right, where we have the estimates of the speciation rate being a lot higher when the trophic level is low. And of course, the trophic level, the super low trophic level, is just fruit, 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 or nectar, nectar, nectar. And the higher trophic level are when we get into insects, or we get even higher into frogs or other bats, or even some birds in some cases. And this is related to their morphology, which I haven't shown. Well, I actually showed the picture of the skulls, but basically the same pattern, where the actual morphology that is a blunter face, a blunter, shorter face, has a higher rate of speciation than the other more square faces. So this is a story about persistence. Because for the longest time, it was actually really hard to grasp how this ecology related to that. But thanks to a partnership with Betsy Dumont, who is pictured here, we were able to answer mechanically. So Betsy Dumont does this really elegant biomechanics, where she created an engineering model of the phylostomid skull so that we could have this diversity that exists in nature and we could study the engineering stressors. How does this skull actually function? We see two different skulls portrayed here. The really blunt one is at the bottom, and a more regular sized one is at the top. But this is diversity. The thing about the phylostomid bats and the Noctilionoid bats is that they encompass almost the entire range of diversity of what the palate looks like and what the skull looks like across all vertebrates. So when did they get so different that mechanically they end up having very different properties? The one that's shown here, A, is more elongated and it's more associated with nectar feeding. The one in the middle is more of a generalist, and C is a frugivore. So when did this happen? Is this an event that happened recently and corresponds to the time when we see that rise in the speciation rate? Or is this an ancient event? How did this diversity come to be? And this is the moment when we get to watch a bat video, because the vast majority of bats, including 100% of the bats that live at this latitude, they hunt insects this way. So I want you to pay attention to what this bat's doing. So the bat, in between its legs, it's got a uropatagium. And the uropatagium is a flap of skin, and it's using that uropatagium and catching a moth and then bringing it up into its face. Right? And so this is called aerial insectivory. And so the vast majority of bats are doing some version of this. But Noctilionoid bats are doing something different, which is dramatically illustrated in this video. So in this video, what we're seeing, this is a fishing bat. And what this fishing bat is doing, it actually has this amazing specializations where it's using its echolocation to search for ripples on the water. And then using that information, extends its long legs into the water and gleaning it up. So Noctilionoids, the vast majority of Noctilionoids are gleaning, including insectivorous Noctilionoids, which includes a whole bunch of phylostomids. All of them are gleaners. Actually, all of the phylostomid insectivores are gleaners. There are some aerial insectivores. We won't get into that right now. But they're gleaners. What do? They look for insects on the leaves. So they go and they do the same thing that this Noctilio is doing on the water. They do it, except they're doing it off a leaf. And so the diversity of ecologies, the diversity of species is more recent, and it's associated with actually being able to generate a new shape of skull. But the diversity in the ecologies is quite ancient. It dates back to the origin of Noctilionoids. And we have proposed that what is going on is that once you are catching insects, instead of catching them on the wing, you are catching them off the surface of a plant. Once you have that ability to do that, you start getting into contact with the plants, and that partnership with plants begins. And this relationship between bats and plants is what's been driving the evolution of a huge number of species diversity, including this Artibius jamaicensis, or the Jamaican fruit bat, which is actually, it's called Jamaican because it was described from Jamaican specimens, but it's actually ubiquitous throughout Central America, Mexico, and most of the islands of the Caribbean. You can see what it's doing here. It's going straight for the fruit. So this is a story of persistence, of actually having this really important question of when and how this diversity is emerging, and persisting through time until we actually had the ability to dive into the past using engineering models and come to terms with when that diversity would have come about. So I'm going to try to close off talking a little bit about some of the things that these bats can do that we really can't. And this is a story, on a personal level, it's a story of resilience. So I'm here because I love bats, and it's a powerful motivation. This is one of those gleaning insectivores that I talked about. It's a beautiful bat. This picture is from Costa Rica. And what they do is they go and they catch the insects from the leaves. But to be successful as a scientist requires for us constantly searching and constantly competing for funds, right? So I'm going to bring a little bit of a downer of a quote, right? And the downer is this: any life, when viewed from the inside, is simply a series of defeats, which is from George Orwell. And so I'm going to share a story about funding, about searching for funding, right? So some 10 years ago, I was in the process of, I was already at Stony Brook, and I was in the process of undergoing a review that is done for people to see if they wanted to stay within their department or not. I actually had already maybe I was already done with the process. It doesn't matter. What matters is that I was applying for this very prestigious source of funding from the National Science Foundation. Now, that's a huge responsibility because the National Science Foundation is taking people's money from taxes and giving it to your lab to do amazing things, to discover amazing things, and to help grow the scientists of tomorrow. So it's not a thing that's done lightly, right? So anyway, I was putting together an application, and this is the application for funding that I paid the most attention to in my entire career. So I started by working, I would do things like I was working all the weekends, nonstop for 6 months, and I sent it out to mentors and to friends to read. And I sent it out, with the hope that they could help me shape this into something that was bigger. I had some really interesting comments along the way. I was hoping to cross all the T's, dot all the I's. And I sent it off. It went off, and it went to reviews, right? And so a few months later, when the decision came, more often than not, this request for funding get turned down. So I was used to that. But this particular one that I had put so much time and energy into, it didn't just get turned down. It got turned down, there are 5 categories of ranking that you can get from excellent to poor. My proposal that I spent 6 months working on got 3 poors. 3 different people who read the proposal thought that this was the worst proposal they had ever read. It was like they had read hundreds of proposals. And me, in my entire career, I've been evaluating proposals, and I've been in panels that have evaluated perhaps 500 proposals, and I have only seen 2 poors. But when I submitted mine, I got 3 poors. So things were looking kind of grim, right? I was getting evaluated at my work. I was getting evaluated, and then on the other hand, I was requesting money, and the answer that came back was not just you don't have money, but this is so terrible that we think it's poor. So what do you do? Well, after about a week of moping, there had to be resilience. And this is a story of resilience, because what you're about to see are a couple of avenues of research that I took up after feeling like the rejection was so profound. I decided that an entire career that had been spent making phylogenies was over, and I was now going to do something different that wasn't making phylogenies after all that time. And that's when I discovered that these bats can do anything. So this is a book from a couple of years ago that I helped edit, that I co-edited, and that summarizes a lot of that research about phyllostomid bats. So what can these bats do, these phyllostomid and Noctilionoid? Well, they can perceive light that is in the blue ultraviolet range, which is pretty crazy if you think about it. These are nocturnal animals. They can find fruit in the dark by scent, and they can do something we can't do, even though we try, which is feed exclusively off nectar, right? So let's see what those look like. So the blue cones are shown here in the purple. So what you're seeing here are the long and middle wave cones in the retina of these bats. And those are in green, and in purple are the blue cones. And you can see that Phylonectes poeti has a lot of them, right? And then the third picture is the 2 of them combined. And what we found was that the blue cones of the bats are actually present disproportionately in the fruit-feeding species. So we don't know the biology of what they're actually doing with these blue cones, with the light that they're perceiving that other bats cannot perceive. We don't know that, but we find this pretty intriguing because it's telling us that fruit feeding has something to do with this perception. And bats also use their sense of smell to find fruit. People already knew that. They had done research on it. You can actually use branches, or you can actually do experiments in the field to get them that they use it to search, explore, and approach when the fruit is ready, right? And so we were able to do, teaming up with a series of partners, we were able to demonstrate that depending on how specialized the bats are, right, they have a greater or smaller diversity of olfactory receptors. Olfactory receptors are really fascinating. Should I be looking at these raised hands? Should I be looking at things that are happening over here? I don't know.
No,
don't worry about it. Yeah.
Okay. All right, just checking. Okay, so olfactory receptors, like you and me, the single largest family of genes in our protein coding genes in your genome are olfactory receptors. We have hundreds of them. Some animals like elephants have thousands of them, and bats have several hundred of them. So we're able to demonstrate that a specialist shown here, up here, Carolia castanea, actually has a lot of olfactory receptors, but they're all of the same kind. Whereas a generalist, which is Carolia perspicillata, actually has few of each kind, but it's got greater diversity. And you can see that in this picture in D, what you see that castanea, when you look at the diversity, is lower, but perspicillata, which is a ubiquitous bat that is found throughout Latin America, it's got this way higher. And then the last thing that we were able to demonstrate that these bats are able to do is feed off nectar. Now, nectar feeding is actually really hard. Like how organisms, including, okay, so here's the second bird that you'll see, and it's a little silhouette of a hummingbird, right? So looking at genetic changes, because the experiment in humans on feeding off primarily glucose and fructose, it doesn't go well. It makes us sick. It causes all kinds of metabolic diseases. And yet, bats, nectar-feeding bats, two subfamilies in the Phylostomidae, Loncophilinae, which is shown in black, and Glossophagiinae, which is shown in the sort of brown, right? They are like hummingbirds. They feed primarily, they're disproportionately feeding on nectar. This is where they're getting their calories. So how do they manage to feed off this? And the answer is, I don't expect, there won't be a test about this. So I don't expect everyone to memorize what's going on here. But I'm pointing out two enzymes that are modified, right? One of them is aldolase that I'm pointing at the top. And the other one is pyruvate dehydrogenase, which is the one at the bottom. And these two enzymes are modified in these nectar feeding bats in such a way that we can actually take the enzymes out and look at their functionality, right? And so when you look at the pyruvate dehydrogenase, it actually has a much greater, a faster function in the nectar bats. And we can see this in the difference between insect and nectar, fruit and nectar, and fruit and insect. You can see that there's not a lot of difference. It's close to zero between fruit and insect, and between fruit and nectar. But between insect and nectar, the action of the enzyme is very different, with the insect minus nectar being negative, which means that the nectar is a lot faster. At the top right, you can see the aldolase activity, right? And once more, we see that the activity in the nectar feeding bat, which is shown in the light green, is actually faster than the activity in the fruit bat, than the activity in the insectivorous bat. So we see these differences in enzyme activity that we can actually replicate outside of the bat, to demonstrate that these genetic differences that we have discovered by comparing their genetic mechanisms have a functional implication that enables them to process the sugar a lot better than we process it. So this is a long-winded way of saying that phyllostomid bats are the coolest, like in this meme where we have phyllostomid bats and all other bats. So I hope that I've been able to share with you a long journey with really cool bats that has included moments of serendipity, persistence, and resilience. And with that, I would like to thank you and all the lab members that make this research possible. Thank you.
Thanks so much for certainly covering a lot of ground and a lot of food for thought. We'll start with, there are some questions, and one is, as species that go extinct on the mainland may persist on islands. Is this true of bats in the Caribbean?
It's mostly the other way around. There are some species that have gone extinct on islands that persist on the mainland. So, for example, there are fossils known from Jamaica of insectivorous and carnivorous species called Tonacia saurophylla, and Tonacia saurophylla is found on the continent, but not on the island. There's another species called Mormops megalophylla, which is a mormopid, and there are fossils that are distributed in the Gulf Coast, actually in Florida. There are some fossils, and in the islands of the Caribbean, but they are no longer present there. They're only present on the continent. So we have examples of the reverse, that species that are persisting on the continent that have gone extinct on the islands, rather than the opposite.
Mm-hmm. Mm-hmm. Another question is, can you say that noctilionoids are generally smaller than other phyllostomid bats based on the skulls?
That's a really good point. So the non-phylostomid noctilionoids, let's remember that the phylostomids are noctilionoids, so it's like boxes, right? Like the phylostomid is a smaller box inside a bigger box. But they are smaller on average because the phylostomids include species like Vampirospectrum, which is very large, but they also include a series of frugivores that get bigger, like the big Artibios. They are relatively big and their skulls are bigger. Whereas the non-phylostomid noctilionoids like Mormops and Pteronodus, they tend to be gracile, small insectivorous bats. So I'm going to hazard that on average, although I could actually kind of use data and crunch that and answer that question. But I'm going to go out on a limb here and say that on average, if we took all the species and averaged them, we're actually going to find that the non-phylostomid noctilionoids end up being slightly smaller.
Let's see. Okay. I have one or two questions that occurred to me as well. There probably plenty of them, but one has to do with the buildup of species in the Caribbean, mm-hmm. and you referred to basically the increase. There's obviously extinction, but mm-hmm. then there's the addition of species either by both immigration from the mainland, for example, from Central mm-hmm. America. And by speciation within the Caribbean, say by isolation on different islands or isolation in different parts of an island, and so on. And I wasn't sure which of these is has been the more important source of the buildup of diversity.
That's really a fantastic question, Doug. What we found, so if you remember your MacArthur Wilson Island biography type, we actually found an equilibrium that was given by sink dynamics, meaning that the offset of the extinction rate was mostly through migration. So the rates at which they're speciating are non-zero, they're positive, there's speciation happening, but they're not enough to offset the extinction mm-hmm. rate, and instead most of the offset is coming from the periodic arrival of new species onto the islands. Obviously, this is happening over millions of years. This is a very long process, but those are the parameters that we get from such model.
Right. And presumably, the on any one island, mm-hmm. The input of species could be immigration of new species from the mainland, let's say, but it could presumably also be by the migration of other populations of the same species that have mm-hmm. survived on neighboring islands. Can you have any handle on that? The extent to which within the Caribbean archipelago, mm-hmm. how important is dispersal from one island to another in maintaining the diversity on your average island, so to speak?
So this is actually a really good question. A model like the one I presented is not very good at answering this because it's just taking things at the species level and it's painting with a very broad brush of evolutionary time. But the Caribbean islands, and I kind of wish I had shown a little bit of that, they are blessed with literal buckets of bat fossils, right? So at the Museum of Natural History in New York, there are layers that are called bat layers. and lizard layers. So if you dig in the Caribbean, yeah, I know, they're all from like the Quaternary. So their entire, you can go Natural History Museum of London, you can go back to the Dominican Republic, and there are buckets of these fossil bats. And thanks to those buckets of fossil bats, we know that some populations of species, that process that I was just talking about, where they go extinct or extirpated and then recolonize, there are some species that do that all the time. Like the Brazilian free-tailed bat or the Mexican free-tailed bat, Tadarida brasiliensis. We find it on islands where it's not present now, but we find it colonizes and recolonizes and colonizes and recolonizes, and it becomes extirpated and it comes back. So there are some species that are able to do that. There's more than that. There's a bunch. I know that, for example, there are still lots and lots of things to discover about the bats of the Caribbean, even though I spent so much time there, and there's a lot of labs now are looking into it. I just heard from a collaborator of mine that a widespread species that everyone thinks is the same thing because they all look the same, it's apparently genetically really different from one island to the other, even though they're capable of flying to one side to the other. So we have examples over and over where once we pair the fossils with the genetics of the species, we discover that the continuity, at first glance, there's a continuity of population that's been there the whole time, but in reality, it's subject to this recolonization dynamics from larger populations on another island or from the continent.
Right.
Yeah.
Encourage more questions. If anyone has any questions about bats at all, now's your chance to get the answer. But in the interim, I have another question, and as you I think remarked at some point, I've been interested in processes of speciation and the role of geographic separation and the role of ecological differentiation among populations, and whether those populations become different species, in part because they become ecologically adapted in different ways. But they aren't mutually exclusive. And I would think is for mammals, certainly for birds, but in mammals I would think as well that populations of the same species become isolated, and that isolation enables genetic differences between them to accrue, including those differences that may lead them to be somewhat different from one another ecologically, to become more specialized on one fruit, like the Carolia bats that you've mentioned, or versus a different fruit or whatever. And so to what extent do you find that that closely related populations on different islands are ecologically different from one another in their feeding or other ways?
So one of the reasons why, and I ended up not showing it, I don't remember exactly why, but one of the reasons why I became intrigued about the Caribbean is that when you go to each one of the islands, the big islands especially, they kind of have one of each, right? All of them have like a fishing bat. All of them have like Artibius jamicensis, which I showed. And they have like one nectarivore, or they may have another nectarivore. And only the largest islands have like two nectarivores, right? So it just seemed like they had these bins, right? These ecological bins where they would fit. But unlike of course, you're familiar, and I don't know if this audience has seen the work of coming out of Rob Losso's lab, where they have this beautiful diversification of the Anolis lizards, right? So in the Anolis lizards in the Caribbean, it's a completely different story. It seems like they have those bins, but they're all from the same common ancestor, right? For the bats, we have a completely different story in which the bins exist, but instead of coming from a single ancestor each time getting colonized, these are bats that get around. The ranges are actually on average larger than the ranges of the average bird, passerine rather, the average passerine. So they end up having these larger ranges. And so what ends up happening is that they fill those bins by being the same species and maintaining gene flow from one island to the next. But ecologically, it's very clear that these niches exist, right? The niche for the nectarivores, and the niche for the frugivores, and the niche for all of that. So we don't have a lot of evidence that's going to drive us to say that they diversified ecologically in the same place. And it's more a process. You're absolutely right. I kind of made it very extreme, like they're mutually exclusive, and they're really not. You can totally have a combination of the two. And what we are seeing is, or the best explanation that we have is that there is this huge ecological diversity that builds up, right? And includes a geographical dimension. So it's not perfectly aligned one thing with the other, but it's ancient. And then they colonize, they disperse, and then fill those niches. I don't know if that fully answered your question. Yeah. I see some new questions are popping up.
Yeah. What is the most important cause of bat extinction?
That's a very, very good question. I think in the Caribbean, one of the arguments that we've had about those species that we see that went extinct before modern times, let's say, but by human hands. So over long evolutionary times, there's processes, predation, etc. That may play some role, but in the human intervention, contemporarily today and in the recent past, it's the habitat change, right? So in the Caribbean, unlike some other parts of the world, people weren't hunting the bats. We don't have any evidence that this is what they were doing. But their habitats did become smaller, right? Now, in present day, the Caribbean is kind of really interesting because Puerto Rico is one of the places in the world that has undergone what's called that forest transition. What the forest transition means is that we hear a lot about deforestation here and deforestation there, but in Puerto Rico, deforestation peaked and now it's declined and it became reforested. Large parts of Puerto Rico became reforested as agriculture was abandoned. So it's one of a bunch of places in the world that have gone in the opposite direction, where there is less intervention of the human habitat and more reforestation, more growth that has come in. And so we see these dynamics that the habitat loss is no longer as important in a place like that. But then we have situations like in Jamaica where we have the feral cats. And so I would say that in present day conditions, one of the things that's happening in a place like the Caribbean is that we need to get a handle on this human introduced feral predators, because these human introduced feral predators are having a huge impact on the populations. When they can roam free, they can go into caves, they can get the pups, they can get, a population like the one Jamaican flower bat. It's got 200 individuals. A couple of cats can do away with the population. So the answer is it depends on the place and on the time, but habitat, I would put habitat destruction pretty high. And now this introduced predators, cats especially.
Which, of course, is the story on islands throughout the world. Cats, introduced predators, cats and rats being the worst of them, are causing extinction of many birds and just about everything else, reptiles and everything else.
Absolutely,
someone asks, are there more ways that bats get sustenance than just gleaning and wing capture that we saw this evening? What about sonar?
That's a really good question. So the sonar is being used in both cases. So when they're gleaning, when they're actually, for example, Noctilio is using its sonar to bounce back the echo.
Remind us what Noctilio is. That's the
fishing bat. The fishing. So remember the fishing bat in the video. So the fishing bat is using the sonar, and it's bouncing the sonar back off from the surface of the water to get that ripple, to perceive that ripple. And using that sonar, that's how it's doing. The aerial insectivore is using the sonar to bounce it off the insects, right, and go catch it. And the gleaning insectivores that are off the leaf are also using their sonar. So sonar is a critical component of all the forms. The only ones that almost don't use any sonar are the pteropodids, which are include the flying foxes. They're not present on our continent. They're not present in the Americas. So the sonar is a critical part of it. There are within this kind of I've painted a very broad brush. Within these big categories, there are subcategories that people who are specialists on that, on basically insectivory and the many ways that it can form. One of my favorite ones is a Noctilionoid bat that's called Pteronodus parnelli, the mustache bat. And what's fascinating about it is that it has evolved here in the Americas, constant frequency collocation with Doppler shift compensation. And that's a mouthful, but that mouthful just means that it's beaming a giant loud sound at a frequency, like a giant wall of sound, that enables it to perceive the wing beat and separate it from other like branches and leaves that are bit, perceive the wing beat. And then the cochlea has what is known as an acoustic fovea, meaning like the fovea in the eye, it's overdeveloped to perceive not the giant sound that it's making, the giant scream that it's making, but the bounce back from that. And it becomes specialized. And when it perceives that bounce back, it performs physics that we don't normally perform to calculate the speed of the insect, independent of, and calculate it as it's flying, to go zone in on a moving insect in the middle of a cluttered background, right? And now this has been evolved by bats in the old world, right? And it constitutes a big family of bats, the rhinolophids, right? And then here in the New World, there's only one species complex, which is a fancy way of saying that there's a bunch of species there and we don't know quite how to divide it, but roughly 10 species, they're all very similar to one another. And they're called the mustache bats, and they're amazing.
And,
there's more questions.
Does weather in the Caribbean, such as hurricanes, cause extinctions and recolonizations?
Oh, what a great question. Okay, so I didn't talk about them because I was just talking about Noctilionoids mostly and phylostomids today. But I studied another group of bats that are called natalids, and they're also a small family that kind of moves very slowly. And they, together with a bunch of other bats, they tend to roost in caves. And one of the hypotheses that at some point I put out there was that this disproportionate cave roosting, which is not very common on the continent at all, it could be the result of hurricanes. It could be the result of this huge disturbance that tree specialized bats in the Caribbean, it's not a good way to be. It really does put you on track to extinction, because even without human intervention, the smaller islands can get completely wiped out, whereas the caves are protected, right? So it's a hypothesis out there. It's definitely something to think about. So hurricanes do have a huge effect. There is some evidence that some hurricanes can actually help move individuals around, interestingly enough. So they play a role in the ecology of the Caribbean. They have played a role in extinctions for sure. And they probably play a role in shaping an ecology where the caves become super, super, super important, and where the bats shape the caves. Yeah. So that inferno, so I showed the Inferno Passage in Jamaica. These caves are called hot caves. In these caves, the bats have created an ecosystem that doesn't exist without the bats. They are in such large numbers, and the cave has to be enclosed just so, and it's got to have humidity. The humidity rises to 95, 99%, and the temperature goes up to 100 degrees inside, just through the heat of their bodies. So the bats have created their own ecosystem in the caves. Yeah.
That's pretty amazing. I think, let's see, you've already talked about how nectar-feeding bats do use sonar. What methods are used to manage the feral cat populations in the Caribbean?
Yeah, so one of the methods that's being used is using exclosures from the roosting caves, right? So trying to put exclosures is not always easy. That they can be managed through the exclosure. That's a current management tool that's being used for the feral cats. There are no projects, unfortunately, unlike places like New Zealand or some of the islands of Australia. There are no projects to eliminate the predators, right? And it's not just feral cats. We're talking about mongoose as well, which was introduced. There's a series of predators that were introduced that are having an effect on the species, on the local species.
Liliana, there are no more questions at the moment. Let me just ask one last question, which is, we don't go. Most of us who live in New York and Long Island and so forth don't go crawling around in caves very much, and we don't get to the Caribbean. If people are interested in, just experiencing bats the way so many of us, spend time birding, how could people in New York enjoy the experience of, how can they experience bats?
How can you experience bats? Okay, so there's this cool tool that you can use that I use sometimes. That's called, it's called an ecometer. And I have an app here for the ecometer. It's called eco, like an echo, and meter, an ecometer. And the ecometer, you can plug it into your phone, and it enables your phone to capture the echolocation calls of bats that are near you, right? And it's got an artificial intelligence module that takes the sounds that the bats have made and estimates with some probability, and it's very good for this part of the world, which species you are looking at. So if you're seeing bats, so for example, here on Long Island where I live, it's a little late for that particular bat, but there's a big brown bat that emerges, especially in the early part of dusk, right? And I can perceive it, and then I can actually perceive its echolocation call thanks to the ecometer, which is an ultrasound. Right. There are a bunch of others, but this is the one that I know that's commercially available that you can just plug into your phone.
Mm-hmm.
Yeah.
Mm-hmm.
So
Super.
that's one way. There's this reserve. There's places where bats roost. There is, thanks to Bat Conservation International, which I encourage you to visit their page and see what they're up to. Bat Conservation International, I've actually recently learned about bat boxes, the installation of bat boxes. This is a question that I often get, and I myself, I'm not an expert in bat boxes, but they actually were able to teach me how you can, like what to look in a bat box, how to make a bat box that's not going to become a bat trap, right? How to make a workable bat box that is going to provide roost for bats and it's not going to cook them or freeze them, which is the problem with some bat boxes, like where it needs to be, what many different kind of models. So if you ever have questions about that kind of thing, I direct you to Bat Conservation International because they know what they're doing. Yeah.
Super.
Mm-hmm.
Thanks so much. Debbie, do you have any last words before we sign off for the evening?
This was a fascinating presentation. So thank you so much for being with us tonight and being so patient and answering all the questions. But we really learned a lot. Thank you.
Awesome. You're welcome. Thank you for having me.
Yep.
Okay.
Thank you, and thanks to all the participants who are still here. Thanks very much.